In this video, we will discuss about Glycan binding proteins. So far, in many of our previous videos, we have understood how this Glycan binding protein interacts with the glycans and cause immune response. Sometimes, which is in favor of the host, and sometimes it is in uh, favor of the tumor cells. So we will start with the C-type lectins. C-type lectins are usually used to, its general function is to detect the carbohydrate recognition domain. And here is the list of different cells where these C-type lectins are found and what are their specificity. For example, these genetic cells, the C-type lectins that are found on the genetic cells can detect these high mannose structures or the proteins or glycoproteins that have these high mannose structures. The macrophages has this MBL C-lectins, and dendritic cells has this MR3 C-type lectin. Macrophages has this MBL C-type lectins, and it can also detect the high mannose carbohydrate structures. Whereas for this endothelial cells has this DG signs C-type lectins that can detect high fucose or high mannose structures on the glycoproteins. And for the epithelial cells and interstitial cells has both the things, DG signs and MGL. The epithelial cells has this MGL, can detect terminal galnac and galactose or sialic acids. Any of these carbohydrate structures can be detected by MGL, whereas this langerine, which are usually found on the common immune cells, can detect high mannose, fucose, and sialic acids. And the B cells has this lectin-1 lectin that can detect beta glucan. So, and this is how the different kind of C-type lectins are been observed on different kind of cells and what are their specificities. Next, we will discuss about understand about this galectin binding proteins. It can detect galectin structures. For example, this galectin 1 can detect the polylactosamine kind of carbohydrate structures, whereas galectin 3 can detect poly. It can also detect this polylactosamine kind of carbohydrate structures, and this galectin 4. And however, the structures looks different. We can see that in the galectin 1 can detect the structures that are in dimer formed, whereas galectin 3 can detect the structures that are in pentamer formed, and the galectin 4 can detect the structures which are in dimer formed, which are connected with some linker, and the galectin N can detect the polylactosamine, which is similar to the galectin 4, to that is linked with a linker, and the galectin line can also detect the polylactosamines. In this case, the in general, the galectin 4, 8, 9 can detect the this proline and glycan rich. We can say that this polylactosamine that are linked with this linker peptide, and in general they are looked similar. And where they are usually found, the galectin 4 are usually found in the tumor cells, whereas galectin 8s are usually found in the reproductive system, and galectin 9 are usually found in the also found in the reproductive system. And now we can understand what are the different kind of Siglecs that are found. And we have understood from our previous videos, many of our previous videos, that Siglec can detect the sialic acids. So the Siglec 1 can detect the sialic acids, whereas the Siglec 2 can detect the sialic acids, or Siglec 2 are also commonly known as the CD22, whereas Siglec 1 is known as the SN. And the Siglec 6 to 9s can also detect the sialic acids that are found on the human beings. And the Siglecs 6 to 9s, those Siglecs 6 to 9s can detect the sialic acids that can be detected by Siglecs 6 to 9s are usually found on the CD33 related Siglecs. Sorry, I want to correct my statement. The Siglec that can detect Siglec 6 to 9s are also commonly known as the CD33 related Siglecs that can detect the sialic acids that are present on the humans. This is the correct statement. Okay. And similarly for the mouse also, the Siglec E can detect the sialic acids that are present in the mouse, and these are also known as Siglec E's are also come in the category of CD33 related Siglecs. So I hope you got a basic idea about where are the different kind of glycan binding proteins found and what are their specificity. If you need to understand more about their role, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.